What if I told you that right now, just sitting there listening to this, your body is actually glowing in the dark?
And, not metaphorically.
Right, not like a personality trait.
No, I mean, your physical cells are actively flashing these microscopic particles of light at one another, literally right this second.
It's wild. They're creating this luminous web of communication across your entire body.
And we are not talking about some fringe, new age theory here
For
either. We
sure.
are talking about ultra-weak photon emissions, which is a heavily documented biological reality where living tissue it emits measurable light in the visible and near ultraviolet spectrum.
Which is just the perfect starting point for today's deep dive. Because, man, we are taking on a massive, incredibly complex stack of sources today.
Oh, it's a huge stack.
Yeah, I mean, we are looking at peer-reviewed scientific reviews, highly technical papers on quantum biology, clinical studies on low-intensity light therapy, or LILT, and even these foundational texts analyzing traditional Chinese medicine.
Right. And the mission here, our goal for this deep dive, is to really bridge that ancient wisdom of energy healing.
Specifically, concepts like qi.
Yes, taking qi and connecting it with the absolute cutting-edge science of quantum physics, quantum mechanics, and modern mitochondrial biology.
Because the intention behind compiling this specific research is synthesis, right?
Absolutely. We are moving past that outdated paradigm where conventional Western medicine just, ignores bioenergetic systems completely.
Right, treating them like they don't exist.
Exactly. What these sources reveal is a necessary evolution. It's a direct integration of bioenergetics with physical biochemistry.
So we are going to completely demystify how energy healing works biologically, anatomically, psychologically, and metabolically.
And I think it's important to say, whether you approach this as a total skeptic or, a lifelong practitioner, we are translating these mystical-sounding concepts into measurable, observable scientific phenomena.
Yeah, we're going to look at the literal mechanics of what happens in the human body when energy is transferred.
But to do that effectively, we kind of have to start by abandoning this idea that energy, at least in the biological sense, is just a convenient metaphor.
Wait, it's not poetry.
No, it's not. If we want to understand how qi operates, particularly when we start looking at the quantum fields later on, we must first ground it in the physical body.
So we're transitioning from the abstract concept of like a vital force to the literal microscopic cellular engines that drive human life.
Exactly. We are talking about mitochondria.
The powerhouse of the cell, as everyone learns in high school.
Right, the powerhouses. And since you listeners are, well-versed in the basics of cellular biology, we can skip the elementary stuff and jump straight into the bioenergetics.
Perfect. So in traditional Chinese medicine, there is this foundational concept of yang qi.
Yes, yang qi.
And the texts describe it as the vital warmth, right? Like the active kinetic energy that fuels your metabolism, your immunity, and just your overall vitality.
And what's fascinating is when you look at the thermodynamic and physiological descriptions of yang qi in those ancient texts, the functional parallels to modern mitochondrial activity are they're undeniable.
They really are.
TCM states that when yang qi is abundant, a person experiences systemic warmth, heightened alertness, and resilience to biological stress.
Which maps perfectly onto optimal mitochondrial function. I mean, when mitochondria are operating at peak efficiency, they're driving thermogenesis.
Literally generating heat.
Right, generating heat to maintain your core body temperature. And they are producing abundant ATP adenosine triphosphate, which is the universal energy currency of the cell.
And the clinical presentations of a deficiency align just as perfectly, don't they?
Oh, totally. In TCM, a deficiency in yang qi presents as chronic fatigue, cold extremities, general sluggishness.
And a drastically slowed recovery from illness.
Exactly. I read those symptoms in the sources and immediately just thought of mitochondrial dysfunction.
It's the exact same clinical picture.
Yeah, when mitochondrial efficiency drops, whether we are talking about the effects of aging, chronic systemic stress, or metabolic syndrome, your energy production plummets and inflammatory markers rise.
And you see those exact same clinical symptoms. The cold intolerance, the severe fatigue, delayed tissue repair.
The sources make a really compelling case that these two concepts are basically describing the exact same vital force, just through two entirely different cultural and scientific vocabularies.
And that synthesis becomes even more tangible when we look at how this energy is supposedly manipulated by a practitioner.
Right, because the sources point us to a highly specific anatomical focal point, the Laogong point.
Yes, in acupuncture terminology, that's designated as pericardium 8 or PC8.
And it's located right in the dead center of the human palm, right?
Exactly, right in the center. Historically, TCM considers this a major energetic gateway. It's specifically the primary center for projecting external ki outward from the body.
And the connection to the pericardium meridian is the crucial detail there.
It is, because when we look at the biological energy demands of the human body, the heart has an exceptionally, almost unbelievably high demand for ATP.
Oh, I read this. The heart muscle actually turns over its entire ATP pool approximately every 10 seconds, just to maintain continuous contractile function.
Right.
I mean, that statistic just stopped me in my tracks. The heart is using and entirely regenerating every single molecule of ATP it possesses 6 times a minute.
The dynamic turnover rate is just staggering.
It's insane.
And in the traditional literature, this relentless, rhythmic generation and expenditure of energy is conceptually described as the pulsation of qi.
Okay, but I want to push back on the mechanics of this for a second.
Sure, go ahead.
Because if we establish that yang qi fundamentally correlates to ATP production, and a qigong practitioner, or like an energy healer, is holding their Laogong point over a patient to project energy.
We run into a major structural problem.
Exactly. We cannot possibly be saying that a human being is physically secreting massive chains of ATP molecules out of their sweat glands, pushing them through the ambient air and embedding them into the patient's skin.
No, of course not.
Right, because biochemistry does not support the transdermal transport of massive molecules like ATP in that manner. It's like trying to throw a car engine through a brick wall.
Your reading of the biochemistry is absolutely correct. And, the scientific literature in our stack aligns with that exact skepticism.
Okay.
There is no biological mechanism for a human to extrude literal ATP molecules to fuel another person's cells.
So what's happening then?
Well, what is actually crossing the physical space is not the fuel itself, but rather a highly specific signal. The sources detail a process called signal-induced mitochondrial activation, or SIMA.
SIMA. So the healer is acting as a catalyst, not a fuel pump.
That is the fundamental mechanism, yes. The practitioner is generating a localized bioelectromagnetic or biophotonic field. And that field interacts with the recipient's biological matrix and acts as a trigger. It signals the recipient's own cellular machinery to radically upregulate its endogenous ATP production.
Oh, I see. So the healer is essentially sending a systemic broadcast that tells the patient's own cellular engines to start manufacturing more of their own fuel.
Precisely. They provide the signal, but the patient's body does the heavy lifting of the actual physical healing.
The implications of that just shift the entire paradigm of how we view non-contact therapies.
They really do.
But wait, if they are projecting a signal across empty space, what is the physical medium of that signal? Like, what is actually traversing the air between the healer's hand and the patient's body?
Well, that question brings us to the science of human light emission, which is where the research becomes truly extraordinary.
Yeah, the idea that humans emit light sounds like science fiction.
Yeah.
But the historical evolution of this science in our sources is fascinating.
It really is.
It starts in the 1920s with a Russian biologist named Alexander Gergech. And he wasn't even studying humans. He was looking at the roots of ordinary onions.
Yes. Gervich designed an incredibly elegant, if controversial, experiment.
What did he do?
He arranged 2 onion roots, so the tip of one pointed directly at the side of the other, and he observed that the cells on the side of the receiving root began dividing at an accelerated rate.
Just from being pointed at.
Exactly. And the division was localized exactly where the tip of the first root was pointing.
So he deduced that the first root must be emitting some kind of invisible signal to trigger mitosis, cell division in the second root.
Right. He hypothesized it was a form of ultraviolet light, which he termed mitogenic radiation.
Mitogenic radiation.
Yeah, and to test this, he placed different barriers between the roots. When he used ordinary window glass, which blocks UV light, the accelerated cell division stopped.
Okay.
But when he used quartz, which actually allows UV light to pass through, the cell division resumed.
Wow. But despite the quartz and glass controls, the scientific community largely dismissed his work for decades.
Right. They did. It wasn't until the 1970s that a German biophysicist, Fritz Albert Popp, managed to vindicate Gervich.
And Popp had access to highly sensitive, newly developed measurement technology that simply didn't exist in the 1920s.
Exactly. Popp utilized photomultiplier tubes.
PMTs.
Yes, PMTs, which are capable of detecting single photons of light. With this technology, he pioneered the modern field of biophoton science, proving conclusively that living cells emit what we now call ultra-weak photon emissions, or UPE.
And these biophotons exist primarily in the visible and near ultraviolet spectrum, roughly between, 200 and 750 nanometers?
That's right.
But we can't perceive them with our naked eyes because the intensity is just astonishingly low.
Oh, it's incredibly low. We are looking at emissions on the scale of perhaps 1 to 100 photons per second per square centimeter of tissue.
Wow.
To put that in perspective, it is millions of times weaker than the threshold of human visual perception.
So you require devices like photomultipliers, which amplify these weak light signals by up to 100 million times, just to register the individual photon hits.
Exactly.
And just to be clear for everyone, this is not bioluminescence.
No, completely different mechanism.
Right, we're not talking about the enzymatic reactions you see in deep-sea fish or fireflies, where luciferin and luciferase combine to create a highly visible glow.
Right, bioluminescence is a highly specialized evolutionary trait designed for macroscopic visibility, mating, predation, or defense.
Right.
Biophoton emission is entirely different. It is a universal fundamental property of living metabolisms. These photons are primarily the byproducts of oxidative metabolic processes occurring continuously within the cells.
And the sources specifically highlight lipid peroxidation as a major source of this, right?
I do.
Can we break that down a bit?
Sure. Lipid peroxidation is the oxidative degradation of lipids. Basically, free radicals steal electrons from the lipids in cell membranes, which results in cell damage.
Right, that sounds bad.
It is, but this process, along with the oxidation of proteins and DNA, creates these intermediate molecules that are temporarily pushed into an electronically excited state.
Ah, so they absorb the energy from the chemical reaction, kicking their electrons into a higher orbit.
Exactly.
But they can't stay there forever.
No, they cannot. The laws of thermodynamics dictate they must eventually relax back down to their normal ground energy state.
Right.
And when they make that transition downward, they shed that excess energy. And that energy is released as a photon of light.
So if these photons are just the exhaust fumes of cellular metabolism, the immediate assumption is that they are just random thermal noise, just chaotic sparks flying off the engine.
And that was the prevailing assumption for a long time, but Pop's research actually contradicted it.
How so?
He demonstrated that this light exhibits statistical coherence. It is not random noise. It possesses the physical properties necessary to carry complex biological information.
Which establishes the foundation for understanding how cells use light for intercellular communication. And the origin point for this communication brings us right back to the microscopic factories generating the energy, the mitochondria.
Yes. The mitochondria are the primary generators of these ultra-weak biophoton emissions. The light generation is intimately tied to the electron transport chain, the ETC, which is the sequence of complexes embedded in the inner mitochondrial membrane that ultimately synthesizes ATP.
And the sources indicate that the ETC generates these measurable light emissions most intensely during periods of oxidative stress.
Yes, they do.
But to understand why, we really have to look at reactive oxygen species, or ROS.
We do, because in classical biology, ROS are almost exclusively framed as the villains.
Right. We are constantly told to consume antioxidants to neutralize free radicals and reactive oxygen species because they cause oxidative damage, accelerate aging, and degrade cellular membranes.
Which is true to an extent, but the biophotonics research presents a massive biological paradox here.
Yeah, this was fast.
While excessive, unchecked ROS lead to pathology, a highly controlled burst of ROS, and one specific type of ROS in particular, plays a critical role.
We need to talk about singlet oxygen.
Let's do it. Singlet oxygen is an electronically excited state of molecular oxygen. Because of its high reactivity, it has traditionally been viewed strictly as a toxic byproduct of metabolism.
Right.
However, the recent literature demonstrates that singlet oxygen acts as a crucial, highly targeted signaling molecule.
Let me try a different analogy for this, because the standard fire alarm or alarm bell metaphor doesn't quite capture the chemistry.
Go for it.
It's more like a biological flare gun or a controlled demolition.
Yes.
The cell intentionally triggers a highly reactive, slightly destructive molecule, the singlet oxygen.
Yes.
And that molecule interacts with the unsaturated lipids in the membrane, sparking that lipid peroxidation we discussed.
And that very specific interaction creates the electronically excited states that emit the visible and near-infrared biophotons.
So the cell is firing a literal flare of visible light into the extracellular space to signal the body's repair mechanisms.
That's a great way to look at it.
It's communicating exactly where the stress is occurring and calling for resources.
The mechanism is just so elegant. Oxidative stress is actually the catalyst for the cellular signaling required for metabolic repair.
Wow.
The generation of ROS and the subsequent emission of light is the mechanism bridging bioenergetic therapies and physical biology. Without that brief, localized flash of oxidative stress, the cellular network would remain blind to the site of the damage.
That makes so much sense. So if the body utilizes light and controlled oxidative stress for internal repair signaling, the logical next step for modern medicine is to try and replicate that process externally.
Precisely. And this is where we see the rise of low-intensity light therapy, or LILT.
Right. Researchers sought to understand how the artificial light generated by a clinical medical device compares to the natural light of endogenous biophotons, or the energy field generated by a practitioner.
And LILT utilizes specific wavelengths of red and near-infrared light. This isn't just any light. It specifically matches the spectrum of the biophotons we've been discussing.
So the targeting is highly specific.
Very. Red and near-infrared wavelengths are utilized because they're absorbed by a critical enzyme in the mitochondrial electron transport chain.
Cytochrome C oxidase, right?
Yes, also known as complex 4.
Okay, so when you irradiate tissue with LILT, the cytochrome C oxidase absorbs those photons, which accelerates the transfer of electrons down the chain, ultimately ramping up the production of ATP.
Yes, but the nuance of how it initiates the healing cascade is the truly fascinating part.
Oh yeah, this blew my mind.
The application of LILT actually induces a transient temporary increase in cellular ROS production.
Right. You are treating damaged tissue by hitting it with a light that initially causes more oxidative stress.
It seems totally counterintuitive.
Yeah.
But it relies on triggering a highly conserved cellular stress response. By inducing a brief controlled spike in ROS, the therapy forces the cell into a defensive, reparative posture.
And the literature outlines exactly what that defensive posture entails.
It does.
The cell drastically increases anti-apoptotic activity, essentially shutting down the pathways that lead to cell death.
Right.
It synthesizes heat shock proteins to stabilize its internal structures. And in this brilliant feedback loop, it ultimately upregulates the total production of antioxidants to manage the environment.
You mimic the distress signal, the flare gun, to force the cell to aggressively fortify itself.
That is so cool.
It is. This metabolic stress response is the physiological bridge explaining how external energy therapies result in lasting, measurable biological changes.
And the clinical outcomes resulting from this mechanism are remarkable, particularly in chronic disease management. But the data reveals a critical variable, doesn't it?
It does. The efficacy of LILT is entirely dependent on the cellular redox state of the patient's tissue prior to treatment.
And cellular redox state refers to the balance between oxidants and antioxidants within the tissue.
Correct. Tissues that are currently locked in a pro-oxidant state, meaning they are suffering from high levels of ongoing oxidative stress and rampant ROS production, they demonstrate a hypersensitivity to LILT.
I love the way you put that earlier. The tissues that are the most degraded, the most damaged, are the ones most hungry for the light therapy.
Exactly. Pathological conditions like diabetic hyperglycemia or ischemic environments where blood flow to the heart or brain is restricted force tissues into extreme pro-oxidant states.
Right.
And these are the exact environments where red and near-infrared light therapies show their most dramatic efficacy.
The Al-Watt-Band study in our source stack provides the perfect quantifiable example of this.
It really is a foundational study for understanding the variable efficacy of light therapies.
So they designed an experiment to measure the effects of LILT on burn healing in rat models. They tested various fluences, different dosages of light energy, specifically 5, 10, 20, and 30 joules per square centimeter.
Right.
And in the control group of normal, healthy rats, applying the light therapy improved the rate of burn healing by roughly 5% to 25% across the different dosages.
Which is a statistically significant, clinically positive outcome for healthy tissue.
Definitely. But the breakthrough came when they applied the exact same light therapy parameters to diabetic rats.
Yes.
These were subjects with systemically delayed wound healing and massive baseline oxidative stress. And in the diabetic models, The burn healing didn't just improve by 25%,
Oh, it didn't.
it accelerated by an astonishing 48% to 76%.
The differential is profound.
It's massive.
It demonstrates that the sicker the tissue and the higher the baseline oxidative stress, the more radically the cells rely on light-mediated purinergic signaling to jumpstart their dormant healing processes.
And the researchers drew a direct parallel here. The mitochondrial electron transport chain activity stimulated by the clinical LILT machines appears remarkably functionally similar to the light-based energy healing processes described in ancient practices like key projection.
Exactly. The physiological pathways being manipulated, the cytochrome C oxidase absorption, the transient ROS spike, the subsequent cascade of heat shock proteins and ATP production, they may be identical, whether the photon source is a clinical laser diode or the biophotonic field of a human practitioner.
So if clinical light therapy is this effective, and we know that damaged pro-oxidant tissues are naturally emitting their own biophotons as a distress signal, the obvious diagnostic question is, how can clinicians measure that natural light to pinpoint the therapy?
And that leads directly into the diagnostic applications of photodetection. If the goal is to personalize light therapy, you must first accurately map the body's endogenous light emissions.
Right. We mentioned photomultiplier tubes earlier, PMTs, which amplify the single photon hits.
Yeah.
But the newer technology involves cooled, charge-coupled device cameras or CCD cameras.
Yes. Modern, highly sensitive CCD cameras, particularly when cooled to extreme temperatures to eliminate background thermal noise, they enable the non-invasive, real-time monitoring of oxidative metabolism in living, intact tissue.
There is a specific study by Kobayashi that highlights the power of this technology.
Kobayashi's research team utilized these cooled CCD cameras to image the spontaneous biophoton emission originating from rat brains in vivo.
Wow.
And they weren't just looking for the presence of light, they were mapping it spatiotemporally.
So they correlated the intensity and location of the biophoton emissions directly with changes in cerebral energy metabolism, fluctuations in cerebral blood flow, and the localized generation of ROS.
They essentially created a real-time visual map of the brain's metabolic activity using only the natural light emitted by the brain tissue itself.
That is just, the clinical implications of this are massive.
Oh, absolutely.
By utilizing biophoton detection mapping on a patient's skin, a doctor could non-invasively scan a joint or an organ field and visually identify the precise regions suffering from localized oxidative stress.
Right. Instead of applying LILT broadly across a generalized area and just hoping for optimal absorption, a clinician could pinpoint the exact coordinates where the biophoton emissions, those metabolic distress signals, are highest.
And then they could target the clinical light therapy, specifically to the tissues primed and hungry to respond.
Exactly.
Okay, before we move on to the human element of this, I think it's vital to provide a clean synthesis for the clinical researchers listening who are actively investigating the biophysical mechanisms of energy healing.
That's a good idea.
How do we summarize the biological baseline we've established?
Well, for researchers attempting to quantify these modalities, the core synthesis is this. Ultra-weak biophoton emission is not theoretical.
Right.
It is an objective, continuously measurable proxy for the mitochondrial redox state and ROS generation within living tissue.
Okay.
Because pathological oxidative stress directly alters both the spectral signature and the intensity of these photon emissions, tracking them provides a non-invasive real-time window into cellular bioenergetics.
That makes perfect sense.
Furthermore, because externally applied clinical light, such as LALT, utilizes these identical oxidative and mitogenic pathways to trigger the highly conserved cellular stress response, endogenous biophoton activity serves as the requisite biological bridge for understanding how non-contact bioenergetic therapies interact with and alter human physiology.
That is the foundation right there.
Yeah.
We have established how machines transmit light into the body to trigger healing, and how sophisticated cameras measure the light the body emits in distress.
Yes.
Now we have to cross the threshold back to the original mystery, the human practitioner.
This is where it gets really profound.
Yeah. How does a healer consciously project energy or chi to initiate healing in another person?
Well, we must return to the anatomy of the practitioner, specifically the Laogong point, PC8, in the center of the palm.
Okay, so when a practitioner engages in touch or near-touch healing, we're looking at a highly specific cascade of physiological events.
We are. Modern biology categorizes one of the primary mechanisms at play here as purinergic signaling.
Let's define purinergic signaling in this context. What exactly is that?
Purinergic signaling involves extracellular molecules, specifically ATP and adenosine binding to purinergic receptors on the surface of cells to transmit information.
Got it.
So when a healer's hand physically touches or comes into extremely close proximity to a patient's skin, they are generating mechanical pressure and a localized bioelectromagnetic field.
And that physical or electromagnetic stimulus interacts directly with the patient's outermost layer of skin cells, the keratinocytes.
Precisely. The stimulation causes the patient's keratinocytes to release their own internally stored ATP out into the extracellular space.
Okay, this circles back to our earlier point perfectly.
It
The
does.
healer isn't giving the patient ATP. The healer's energetic presence acts as the precise stimulus required to force the patient's own skin cells to dump their endogenous ATP into the surrounding tissue.
Exactly. And the sudden presence of massive amounts of extracellular ATP initiates a profound purinergic signaling cascade.
Which does what?
This cascade propagates through the local neural pathways, resulting in rapid pain reduction, vasodilation, and the acceleration of localized tissue repair.
Wow.
By focusing their intent and physiological energy through the Lagong point, the practitioner is essentially optimizing and directing this native biological signaling pathway.
That beautifully explains touch and near-touch healing. The physical proximity triggers the keratinocytes.
Right.
But, the sources push us into territory that completely breaks that model.
You do go much further.
Yeah, they extensively document instances of intentional energy projection where the practitioner is actively fighting complex diseases like cancer from a distance.
The research surrounding external qi, or EQ, projection is rigorous. Specifically, the studies involving Dr. Yan Xin, a highly credentialed Qigong master.
Right.
Researchers have spent years exploring the measurable links between a practitioner's mitochondrial ETC activity during focused intention and the resulting biological effects on isolated tissues.
And the results of the Yan Xin studies defy classical biological expectations entirely.
They really do. The studies consistently observed that the projection of external chi selectively inhibited the growth and proliferation of malignant cancer cell lines.
We're talking colorectal, lung, and pancreatic carcinomas, right?
Yes.
The mechanism of action is what fascinated me, though, because it didn't just indiscriminately kill cells.
No, it was highly targeted.
Right. The external key induced apoptosis, which is programmed cell death, in the tumor cells by downregulating very specific survival pathways.
Namely, the ACT and ERK12 kinase cascades.
Yes. While simultaneously modulating NFKB, which is a protein complex that controls transcription of DNA and cell survival.
By altering the phosphorylation state of these specific kinases, the energetic intervention literally signaled the malignant cells to dismantle their own structure.
That's incredible.
But the critical defining characteristic of these studies was the selectivity.
Right. While it was triggering apoptosis in the carcinomas, the exact same external key exposure actively protected normal, healthy cells like fibroblasts and cortical neurons from induced oxidative stress.
It shielded the healthy cells while systematically dismantling the malignant ones.
It even demonstrated the ability to restore the functionality of crucial enzymes, like PI3K, that had been chemically deactivated.
Which is just staggering.
But here is where the fundamental paradox arises for me.
Okay, what is it?
If purinergic signaling requires mechanical pressure or a highly localized electromagnetic field to stimulate the keratinocytes, how is Dr. Yan Jin affecting isolated cells in a petri dish?
Right.
More profoundly, how do the sources document practitioners effectively altering biological markers in patients located across the room or in some cases miles away?
This is the exact threshold where classical Newtonian biology fails us, and we are forced to enter the quantum field domain, or QFD.
Okay, unpack this.
Well, from a classical physics standpoint, the inverse square law dictates that any electromagnetic field degrades exponentially over distance.
So distance healing should be impossible.
Under classical physics, yes. But the observed efficacy aligns perfectly with the principles of quantum mechanics.
How does quantum mechanics bridge a physical distance of miles to trigger apoptosis in a cancer cell?
It relies on the concept of quantum entanglement.
Oh.
The observation that vast physical distance does not diminish the efficacy or the targeted specificity of certain energy therapies heavily suggests a quantum-like entanglement occurs between the practitioner and the patient.
So, if the biophoton fields we discussed earlier are involved in this interaction, you are suggesting the healer and the recipient effectively merge into a single coupled quantum system.
Biophysically, yes. The practitioner engages in specific meditative and breathwork practices to cultivate a state of extreme energetic coherence.
Right, to maximize the efficiency of their own mitochondrial ETC and achieve a high ATP phosphorylation state.
Exactly. And in an entangled state, this biological coherence becomes non-local.
Non-local meaning the physical space separating them just becomes totally irrelevant to the transfer of information.
Precisely. The patient's cellular network, operating within this coupled system, is able to effectively borrow the energetic coherence and the low entropy state of the healer across space and time.
To facilitate necessary repairs and upregulate their own depressed pathways. I'm trying
Yes.
to visualize the mechanics of this, though. How do subatomic particles inside my mitochondria borrow energy from a practitioner's mitochondria located in another city?
To conceptualize that, we have to look at the properties of the zero-point vacuum and the phenomenon known as quantum tunneling.
Okay, quantum tunneling.
In classical physics, if a particle needs to cross a barrier, say an electron moving through a complex in the mitochondrial membrane, it requires a specific amount of energy to physically breach that barrier.
Right.
If it likes the energy, it simply cannot pass.
It's the classic analogy of rolling a boulder up a hill.
Yeah.
If you don't impart enough kinetic energy, the boulder stops halfway up and rolls back down. The barrier is insurmountable.
Exactly. But at the quantum level, particles like the electrons and protons moving through your respiratory chain do not behave like solid boulders.
They exhibit wave-particle duality, right? They act as probability waves.
Yes. Quantum mechanics dictates that even if a particle does not possess the classical energy required to surmount the barrier, there remains a distinct, calculable probability that it can simply tunnel directly through the barrier and appear on the other side.
It effectively bypasses the physical limitation of the barrier entirely.
Yes. And the probability of that tunneling occurring can be externally influenced by fluctuations in the quantum field domain.
Okay.
Because the zero-point vacuum is not simply empty space. It is a highly dynamic, structured medium teeming with virtual energy fluctuations.
So when a practitioner projects chi, they are interacting with this zero-point vacuum.
The hypothesis suggests that a highly trained practitioner establishes a bioresonance between their coherent biofield and the patient's biofield. And this resonance is mediated instantly through the underlying quantum vacuum. This non-local resonance actively alters the local probability field surrounding the patient's cells.
So it facilitates and drastically increases the rate of quantum tunneling within the patient's mitochondria.
Exactly. It instantly optimizes their electron transport chain and ramps up ATP production without a single physical molecule needing to cross the geographic distance between them.
Wait, so the practitioner is not sending energy. They are restructuring the localized probability field of the patient's quantum environment to ensure the patient's cells function at maximum thermodynamic efficiency.
That's it exactly. They are acting as a quantum catalyst. They modify the probability structure of the local field, and the biology follows the physics.
The implications of that are just staggering. But, to validate that this is not simply a highly complex theoretical framework, the sources demand we look at how intention directly and measurably alters physical biology.
We do.
And to eliminate the variables of human belief and the placebo effect, researchers started by examining organisms much simpler than humans. Microbes.
Yes, because if coherent biophotons act as a universal communication system mediated by quantum principles, we should observe this phenomenon in the simplest biological models.
Right.
And the literature provides that evidence. Researchers have extensively documented intercellular communication mediated exclusively by biophotons in isolated microbial populations.
The sources detail the Grasso experiments from 1991, which are incredibly compelling.
Grasso's methodology was brilliant. It was designed to isolate optical communication.
What did he do?
He utilized Saccharomyces cerevisiae, which is just common baker's yeast. He separated two distinct populations of yeast using physical barriers that blocked chemical transfer but permitted the passage of visible and UV light.
So the two populations of yeast could essentially see each other, but they could not chemically interact or share nutrients.
Correct. And Grasso demonstrated that the biophoton emissions from an actively growing population of yeast were capable of inducing significantly higher rates of gemme formation.
Which is the process of asexual reproduction and cellular budding.
Yes, exactly. It induced that in the secondary separate yeast population.
So the light flashes from the thriving yeast, literally signaled the neighboring colony to accelerate its own growth and division.
With no chemical contact, only an optical exchange of biological information.
The yeast cells are utilizing an innate optical communication network.
They are.
And if a simple single-celled organism like Saccharomyces cerevisiae utilizes this mechanism, it is entirely logical that the highly complex trillion-cell network of the human body utilizes a massively more sophisticated, integrated version of this exact same biophotonic communication system.
Absolutely. Which brings us to the profound challenge of objectively measuring this invisible energy transfer between a human healer and a patient.
Right. How do you construct an experiment to capture quantum bioresonance?
It requires extreme environmental control. The sources outline methodologies utilizing dual PMT walk-in dark chambers.
So they build a completely light-proof, pitch-black room and equip it with incredibly sensitive dual photomultiplier tubes.
Yes. You place both the practitioner and the patient inside the zero-light environment. The goal is not just to measure if light is emitted, but to look for temporal photon coherence between the two individuals.
Okay, so the researchers are utilizing the dual photomultipliers to determine if the specific rhythms, spikes, and valleys of the biophoton emissions from the healer begin to temporally synchronize with the emissions of the patient during the active healing session.
Exactly. They're looking for visual proof of that entangled, coupled quantum state.
But if the healer is doing this profound level of catalytic work, altering probability fields and emitting coherent light, they must be expending their own metabolic fuel to maintain that state.
And the metabolic expenditure is highly measurable.
Oh, really?
Yes, the research demonstrates a clear psychological and metabolic interaction during energy projection. A specific pilot study measured the baseline biophoton emission from the hands of experienced healers before a session.
Okay, and what happened to their emissions after they finished projecting Ki?
After the healing session concluded, the biophoton emission from the practitioner's hands had diminished by a statistically significant 11%.
An 11% reduction in light output.
Yes.
They literally drained their own cellular batteries to project the field.
It provides hard biological proof that an active expenditure of bioenergy is occurring.
Which just thoroughly dismantles the argument that energy healing is purely a psychosomatic placebo effect within the patient.
The practitioner is physically utilizing their own mitochondrial reserves to generate the coherence, and the connection between the focused human mind and the physical emission of light goes even deeper than that.
We have to discuss the Balkan hypothesis.
The Balkan hypothesis was perhaps the most paradigm-shifting concept in the entire stack of sources.
It really was. The Balkan hypothesis posits that visual imagery, focused thought, and intention actively and spontaneously produce biophotons within the brain tissue itself.
Right. Researchers placed human subjects in entirely dark environments and instructed them to actively imagine a bright light, focusing their intention intensely on that visualization.
And the PMTs measured the result.
The focused intention produced statistically significant increases in ultra-weak photon emissions, emanating specifically from the right hemisphere of the subject's brains.
That is wild. The act of thinking, the act of visualizing a light, literally generated physical photons of light that permeated the skull and were captured by a machine.
It did.
Our conscious thoughts and intentions are physically generating the light that our cellular networks use to communicate.
It forces a fundamental reevaluation of consciousness. It suggests that consciousness is not merely a passive byproduct of neurological activity, but a prime active mover of biological energy flows.
But it is crucial to note that optimizing this system is not always about generating more light, right?
No, definitely not.
The sources detail how certain practices actually lower your overall light output, which is highly beneficial.
That is a vital nuance. We established earlier that biophotons are largely a direct byproduct of oxidative stress, lipid peroxidation, and ROS generation.
Right.
Therefore, interventions that profoundly reduce systemic biological stress should logically result in a reduction of baseline light emission.
Less systemic stress equals fewer free radicals, which means less cellular exhaust lighting up the PMTs.
Exactly. Clinical studies evaluating long-term practitioners of transcendental meditation, or TM, revealed that regular, deep meditators present with significantly lower baseline ultra-weak photon emissions compared to non-meditators.
Wow.
By consistently engaging the parasympathetic nervous system, their bodies maintain a much lower ambient level of free radical reactions.
And they achieved similar results using phytochemicals too.
Yes, specifically utilizing adaptogenic herbs known for their stress-modulating properties.
Like rhodiola, right?
Exactly. A 2009 clinical study focused on rhodiola. Human subjects who supplemented with it for just 1 week demonstrated a significant, measurable decrease in total photon emission compared to a placebo control group.
They actively lowered their systemic oxidative stress through the adaptogen, and the light detectors provided the objective proof of the metabolic shift.
It validates the systemic impact of both psychological and biochemical interventions on the body's biophotonic output.
So, with all of this heavily documented data, the Grasso yeast experiments, the PMT darkroom synchronization, the 11% energy drain in healers, the Bocan intention imaging, the TM and rhodiola results, the inevitable question arises.
Why isn't this mainstream?
Exactly. Why is this framework not currently integrated into mainstream allopathic medicine?
Well, the sources are highly transparent regarding the current scientific and epistemological challenges inherent in documenting the objective effects of external energy healing.
The roadblocks are substantial.
They are.
What would you say is the primary methodological challenge?
The foremost difficulty lies in isolating intention as an independent variable.
Okay, how so?
In a standard clinical trial for a pharmaceutical, the chemical compound is the sole variable. In energy medicine, the practitioner's focused mind, their fluctuating emotional state, their level of fatigue, and the patient's individual receptivity to the biofield are all massive, constantly shifting variables.
So if the practitioner is experiencing personal stress, their quantum coherence degrades, and the clinical result cannot be replicated.
Exactly. It is incredibly difficult to design repeatable, double-blind, standardized trials for a deeply subjective, dynamic, non-local human interaction.
That makes sense.
Secondly, there is a severe technological hurdle.
Separating the biophoton signals from the background noise, I imagine.
Yes, the biophoton signals are so incredibly weak, just a few photons per second, that the slightest environmental contamination, a minute shift in ambient temperature, or minor cosmic radiation can completely ruin the dataset.
Wow.
We are trying to measure quantum effects in a macroscopic, inherently messy environment.
And beyond the technology, there's the philosophical resistance.
The sheer epistemological hurdle. Conventional Western medicine is fundamentally built upon a biochemical reductionist framework,
Right.
introducing a biophysical quantum mechanical paradigm that requires acknowledging non-local consciousness requires a systemic paradigm shift. Many medical institutions are simply not structurally or philosophically prepared to make that leap.
Yeah.
I can see that. We are attempting to measure an interconnected, holistic quantum system utilizing statistical tools originally designed for isolated reductionist variables.
It is difficult to write a prescription for quantum entanglement on a standard pharmacy notepad.
It requires an entirely new language of medicine.
This has been an absolutely mind-bending journey through the literature. We started with the ancient Taoist concept of yang qi and mapped it directly onto the thermodynamic reality of the mitochondrial electron transport chain.
We did.
We explored how those mitochondria utilize ROS and singlet oxygen to fire biophotonic flare guns during oxidative stress. And we examined how both clinical LALT machines and human practitioners manipulate that exact same purinergic signaling pathway to aggressively trigger cellular repair.
And we bridged the seemingly insurmountable gap between physical, localized touch and the non-local realities of quantum tunneling and energetic entanglement.
It really is a profound testament to the fact that integrating multiple perspectives, the ancient energetic and the modern biophysical, deeply enriches our comprehensive understanding of human biology.
I couldn't agree more.
As we close out this deep dive, I want to connect this massive amount of scientific data directly back to you, the listener. Think about your own daily energy expenditure.
Yes.
Consider how your chronic stress levels, your sleep hygiene, and even your moment-to-moment emotional states might be actively altering your internal cellular glow.
Every conscious thought, every physical action, and every emotional reaction directly influences your mitochondrial efficiency and your systemic oxidative stress levels.
Supporting your own yang qi. Whether you achieve that through rigorous exercise, prioritizing restorative sleep, practicing deep meditation, or utilizing targeted red light therapy, is fundamentally about supporting the microscopic integrity of your mitochondria.
Exactly.
You are actively tending to the engines of your own light.
And I would offer one final profound thought to consider based on the Bocan hypothesis and the Yanshin studies.
What's that?
We have discussed at length how highly focused, cultivated human consciousness can direct biophotons and alter probability fields to physically heal tissue from across a room.
The measurable power of intention.
Yes, if that level of targeted, coherent intention is physically real and scientifically measurable, what does that imply about the passive, everyday thoughts, anxieties, and intentions we constantly project onto ourselves and onto the people around us?
Oh, wow.
If the quantum field domain truly connects all biological systems, are we constantly engaged in a subtle, entirely invisible exchange of quantum light and probability without even realizing it?
That's a huge thought.
Does your daily emotional state literally cast a measurable light or a measurable shadow on the cellular networks of the people you interact with every single day?
That is a staggering implication. Are we
Yes.
constantly exchanging light and altering each other's cellular reality? Well, keep questioning the boundaries of what you think is biologically possible, keep exploring the science, and perhaps be a little bit more mindful of the specific energy you are broadcasting into the field today. Thanks for joining us on this deep dive.